Welcome to Chapter 9. So this week we are going to talk about muscles. And the first part of this is to discuss muscle and muscle as a tissue. And then for Chapter 10, we will move on to discussing the individual muscles. So this set of topics, I feel, is the most difficult of the ones we have done so far and the most difficult of the semester. And that's really because this is the first time that we have seen a body system in which we are going to talk about a process and a multi-step process of muscle contraction and how that happens. Action potentials, the movement of ions, we're getting into some biochemistry. And so rather than just thinking about, oh, I have to know that this is the humerus, oh, I have to know that this is stratified squamous, oh, I have to know that this protects me from abrasion. It's much more, it's been up to this point, a lot of memorization, a lot of what is this and where is it and why does it do what it does. In Chapter 9, we're going to talk about muscle tissue from the cellular level and how muscle contraction happens. And that's going to involve structures that you've never talked about before, along with some electrochemical gradients and the movement of ions to generate action potentials. And it's a complicated chapter. So I really recommend that you sit with this one a little bit, read through it, make sure you're watching as many videos as you can so that you can understand the process of muscle contraction. And we are going to spend most of our time talking about skeletal muscle for this chapter. We'll do a little bit of smooth muscle at the end, and we probably won't touch on cardiac all that much. That's because for cardiac muscle, you will talk about that again in A&P2 when you do the unit on the cardiovascular system. So we're predominantly, like I said, going to focus on skeletal muscle contraction with a little bit on smooth. So we're going to start out by talking about how we compare and contrast the three different types of muscle tissue in the body. This little video is talking about treating a muscle strain with RICE. RICE, rest, ice, compression, elevation. So again, check out the video. All right, so muscle tissue. Nearly half of our body mass is muscle, right? We have bone and muscle, the two things that sort of weigh the most, then our abdominal organs from there. But most of our abdominal organs have layers of smooth muscle in them. All of our digestive organs, bladder, etc. The muscle as a tissue transforms chemical energy into mechanical energy. So we're going to take that ATP and we are going to see how that actually causes muscle fibers to cross each other and exert force. So we're going to look at the types of muscle tissue, of which we have skeletal, cardiac, and smooth. We're going to look at the characteristics of each one, right? What does muscle tissue do? And then we're going to look at the functions, right? Obviously, cardiac muscle is for pumping the heart. Smooth muscle moves fluid through a tube, and skeletal muscle moves our bones. So the overview. So the types of muscle tissue, when we're talking about muscle tissue, if you see the terms myo, MYS, or mice, or sarco, right, those are going to be muscle. We have sarcolemma. We have myometrium. We have different words that evoke muscle tissue. All right, skeletal, cardiac, and smooth. All right, so skeletal muscle characteristics packaged into skeletal muscle. So each muscle is an organ. So the bicep is an organ, the tricep is an organ. Organs that attach to bones and skin. Skeletal muscle fibers are the cells themselves. So when we say a skeletal muscle fiber, we mean a skeletal muscle cell. They're voluntary because consciously controlled. I can decide to flex and extend. Contract rapidly, tire easily, but are very powerful. This is important. So smooth muscle is slow, sustained contractions, but doesn't get tired, right? Heart, very rapid contraction, but thank goodness it doesn't tire easily. It happens 24 hours a day, 7 days a week. So skeletal, striated, voluntary. Those are the three things you want to know. We'll talk about what striations are in a minute. Cardiac muscle, only in the heart, so not cardiovascular muscle, just cardiac muscle. Striated, similar to the skeletal muscle, and we'll talk about what that means in a minute. Contracts steady, right? Boom, boom, boom. Involuntary, you don't think about contracting your heart. Then we have smooth muscle, totally different, found in the walls of our hollow organs, not striated. So when we talk about the striations, we don't see that in smooth muscle. Involuntary, right? You don't think about contracting your small intestine to move your food along, right? Doesn't happen. So you can see how we can compare and contrast. Always makes a great essay question or short answer question, comparing and contrasting the three different types of muscle, right? Smooth and cardiac or voluntary. Cardiac and skeletal are striated, right? So we kind of have a good compare and contrast question there. This table 9.3 gives you the location, the cell shape. Oh, there's more. There's more to 9.3. I don't know where the rest of it is. And the function and the contraction and all of that of the different types of muscles. So you can go in and find that in your book or on Mastering. Muscle tissue, no matter what type, has four main characteristics. They are excitable, right? Their ability to receive and respond to stimuli. They are contractile. They shorten when stimulated, right? When I contract my bicep, it goes from being this long, and I contract it, and it becomes this long, right? We're contracting. Extensive, meaning I can stretch it back out again. I can take my wrist and stretch the muscles of my forearm. And then elasticity, ability to recoil to the resting length. So we can contract it, we can extend it, and then we can let it just elastically relax. All right, so four characteristics. And that doesn't matter whether it's skeletal, cardiac, or smooth. They all have those characteristics. What are the functions of muscle? Now we're talking about skeletal muscles. I probably should say skeletal muscle functions. Producing movement, maintaining posture, stabilizing joints, and generating heat. These are the four skeletal muscle functions. Each muscle has a nerve, an artery, and some veins, right? Contracting muscles require a huge amount of oxygen and sugar to make ATP. All right, so we need a lot of vascular support. If you look at the tissue at the bottom right, that's going to be the femur, you'll notice, and it looks like there's some sort of muscle attached there. Maybe it's a quadricep muscle. And you'll see how it starts at the bone, and you see the tendon, and it goes out around sort of the bulk of the muscle. And you'll also notice that the muscle itself is made up of these smaller units called fascicles. And then there's a fascicle made up of these smaller units of muscle fibers. Each muscle fiber, remember, is a cell. So it's the organ itself, the muscle, then made up of fascicles, which is made up of muscle fibers. And even the muscle fiber, you will notice, has what looks like a set of straws. And each one of those is going to be called a myofibril. And we'll get to that subcellular structures in a minute. The other thing I want you to notice about this picture is that muscles are covered with layers of connective tissue called epimysium around the whole muscle, perimysium surrounding each fascicle, and endomysium surrounding each muscle fiber. Now, all of these are continuous. So it starts at one end of the muscle, goes around and through the muscle, and then connects again at the other end of the muscle. So what that does is allows that the entire muscle, whether it be the outside or the inside, is attached at the end to the tendons. And so when the muscle contracts, the whole thing contracts. When the muscle relaxes, the whole thing relaxes. It works as a unit because the connective tissue in and around the epimysium, perimysium, and endomysium allows that the entire thing is attached at both ends. So muscles attach bone to bone, right? And they go sometimes over joints. That's how we're moving our joints. We have the origin, which is the immovable bone, and the insertion, which is the movable bone. So if I'm talking about my bicep and I'm flexing my forearm, I'm closing the angle, right? Flexing. My bicep is doing that. It's attached at the shoulder. That's the immovable bone. And it's attached at the, because it's called a bicep, It's attached in two places, the radius and the ulna, and pulling up on both of those. So this is the movable bone. This is the insertion of the bicep, right? This is the origin of the bicep. All right, so we can have direct or indirect attachments. You'll do more about this in chapter 10 when you're looking at where each muscle is. So now we're going to go down into the microanatomy of a muscle cell. Now, skeletal muscle, like we just saw back here in this picture, we're down to the muscle fiber level. And the muscle fiber level in this picture, you can see little purple nuclei, right? And then all of those round cylinders kind of sticking out. Well, what does that mean? So the long cylindrical cell, around it, we're going to call that the sarcolemma, all right? The muscle fiber plasma membrane. So just like any other cell that has a plasma membrane around it, we're going to call this the sarcolemma. Inside, right, we have glycosomes, myofibrils, just different modified organelles that allow the muscle cell to do what it's supposed to do. So here's a picture of that muscle fiber, that muscle cell, really close up. Now what you'll notice are a couple things. One, that it's peeled back the sarcolemma, that one layer, the plasma membrane. Inside, we have all of these myofibrils. You'll see about 50 of them. You'll see big, dark purple nuclei. And what you'll notice about these cells is that it's multinucleate. There's more than one nucleus in a muscle cell. What else are you going to notice? Mitochondria are in there. And why are mitochondria in there? Because mitochondria make the ATP. And so we need energy, right, in these cells. Now, when we're looking at this, what you'll notice just looking straight on is that it looks striped. We have light, dark, light, dark, light, dark, right? Those are what we refer to as striations. Striations are the dark and light banding pattern that we see on a muscle fiber. And those striations are the interactions between two little microscopic fiber types that we're going to get to in a minute. So this is one picture of a muscle cell, multinucleate, lots of mitochondria, and a bunch of myofibrils. Right, and these are all organelles. Let's go in a little bit further. So here we are. We're looking at the light and dark, light and dark, light and dark. Here we are getting a little bit closer. This is a photomicrograph. This is what it actually looks like under a microscope. And if you're in the lab session, we will look at this in lab this week. So you'll see the purple nuclei. You'll see the red and skinny white bands, right? And they all have names, of course. So the striations are just the stripes themselves. When we talk about the red, that's the dark band. That's called the A band, all right? And then we have the I bands, which are the lighter regions. So if we take more of a cartoon picture of that and get even closer and a little bit more clearer, right? We'll see this is a picture of what's called a sarcomere. And a sarcomere is one unit. Notice how it goes from that wavy Z disc to wavy Z disc. That's one sarcomere. And from there, that's the contractile unit. You can see how the red are like this, and the blue is here. And you can see how if I were to take two ends of that and squish it together, the bands would just go like this, whoop, and then they could come back out. And they could go in, and they could go back out. So you can literally accordion that and watch it go in and out. And that's just contraction at the microscopic level. So the smallest contractile unit of a muscle fiber is called a sarcomere. It has an A band, which again is the red, and then the I band, right, which is the white at the blue zigzag. Now, what does that mean? So let's go in even further. All right, so notice we have red and blue. If we're talking about different types of fibers, red and blue, the lines on that picture. Here we are with red and blue with a little added yellow. So the red here are these bands, and you'll notice they have these little like lumps sticking off of them. That's called a myosin. All right, those are the red myosin thick filaments that extend the length of the A band. So we have this A band and that thick red, there's three of them. The thick red lines are myosin with the little extensions. Then we have the blue. It just looks like twisted blue cord together. There are four of them. Those are called actin filaments. That's the blue. So when we go back here, the blue is actin. The red is myosin. All right. And you'll notice how the red look as though they kind of reach up and around, like kind of extend towards the blue. And we'll see why that's important in a minute. Something that's added in here that we haven't seen before is the yellow. The yellow is an elastic filament. It's called Titin. And Titin is what's going to allow the recoil. Remember when we talked about the functional characteristics? We had contraction, and then we had stretch, and then we had recoil, right? We had that elasticity. Well, that's where this Titin comes in. All right, so this is the red thick filament. It's also called myosin. It has polypeptide chains, and you'll see how it has these extensions called myosin heads. And in the bottom of that image, it says the actin binding site. So when we look at this picture and we see that the red has these little extensions, these little heads, they are actually going to touch that actin filament. All right, so during contraction, the heads of the myosin link together with the actin. Here's the actin. Now you'll notice that the actin has two more yellow molecules in there. But you'll also notice that each of the actin blue dots has a sort of a dark blue area to it. That's the myosin binding site. So these two myosin heads fit into the blue dots on the actin. That's going to be important that we understand this. So we have the actin up here, we have the myosin here, and these guys actually stick on to that actin. Now it doesn't obviously, it's not going to be that easy, but this is going to be actin. The yellow things, the troponin and the tropomyosin, are regulatory proteins. Those are going to regulate muscle contraction, right? We don't want to be constantly contracted. We contract and relax, contract and relax. So that's what those two are going to do, and we'll get into that a little bit. So this is the thin filament of actin with the regulatory molecules of troponin and tropomyosin. It's getting complicated. All right, now, myofibrils. We have the elastic filaments that we talked about. We have dystrophin, which links them together in the sarcolemma so that they don't move around inside the plasma membrane. So all of those are all stacked up against each other, and these actually attach to the sarcolemma, so they all move together. They're not rolling around in there. Then we have nebulin, myomycin, and C proteins that bind the sarcomeres together, right? So we just want to align that. We want to make sure that when we're creating this muscle fiber, that is a long, thin cylinder all in one line. And these other molecules help with that. All right, so this is a picture of 5 myofibrils linked together. And we've added some things. So we can see in the top one, you see the red myosin, you see the blue actin, you don't see the titin, you don't see the tropomyosin or the troponin, but you know it's there. Then we have these other ones, and these are linking together in a group of 5 with that blue stuff and then the white bands that go around everything. And the blue stuff is called sarcoplasmic reticulum. Now we heard about endoplasmic reticulum, or you know about it from your study of cells. And this is sarcoplasmic reticulum. Sarco meaning muscle. So sarcoplasmic reticulum. The role of the sarcoplasmic reticulum is to moderate calcium. I talked to you a little bit about calcium when we did the chapter on bones. And I said that calcium, while important for bone structure, is super important in muscle contraction. And here we are. We got there. The sarcoplasmic reticulum in the blue regulates calcium levels. Calcium levels are vital to the interaction between actin and myosin. We'll see why in a minute. We're still in the microanatomy, for goodness sakes. All right, so we have the myofibrils. You can see some mitochondria in there. You can see the sarcoplasmic reticulum and something called the T-tubule, which is the white. All of this is going to be important later. This is just laying out the framework for where we are. So the sarcoplasmic reticulum regulates calcium. The T-tubules connect, right? The sarcolemma connect those to each other. It says a tube formed by the protrusion of sarcolemma deep into the cell's interior. So we have this entire cell surface, but we want all of the myofibrils, all 100 myofibrils, to be in sync and contract together. If I'm going to flood a surface with calcium and I just flood it out here, only the myofibrils right inside the plasma membrane are going to be affected. So what we want to do is we want to extend that sarcolemma into the middle ones so that everybody can get calcium at the same time. So that's what that white does. It extends from the sarcolemma and deep into all of these smaller myofibrils. All right, so we have a couple of things when we want to, that we need to talk about. So let's think about what happens in muscle contraction. So I think about it. Voluntary. I want to contract my bicep. I want to do this. There are probably 20 steps that just had to happen between here and here. Some of them, most of them are biochemical, and a lot of them are going to be physical cross-bridge activation where the sliding filaments, where those thick and thin filaments slide across each other and shorten the muscle. All right, so the bottom one says when the nervous system stimulates a muscle fiber, the myosin heads are allowed to bind to actin, right, and then they walk down. All right, so we saw those two heads and they bind and bind and bind and they walk their way down the actin. Well, as they walk their way down the actin, what are they doing? Right, they're making that shorter, so we're getting contraction. All right, if you need to take a break, right, push pause, come back, because now we're going to talk soup to nuts, how we're going to get from the nerve all the way to the contraction, and that's going to be quite a thing. So here we are. On the left-hand side, we have a relaxed muscle. On the top is an actual photomicrograph. On the bottom is what's happening with those thick and thin fibers and that tighten, that elastic fiber. When the muscle is relaxed, all right, we have the actin and the myosin kind of far apart. There's a lot of space here. You'll notice in the top picture, there's a lot of white, which means we have a lot of place to shrink down. On the right-hand side, we have a fully contracted muscle fiber. Notice how there's not a lot of white that's squished all the way in. Notice how the myosin and the actin, right, have walked down each other and shortened. On the bottom picture, notice how those eye bands have gotten a lot shorter. But what you'd notice is that the fiber lengths don't change. It's not as though we have these two fibers, these are actin, this is myosin, right? And they shorten like this. That's not what happens. They slide over each other and slide back. So they don't change size, they just slide over each other, become more compacted, right? So that's why it's called the sliding filament model. We're not talking about scrunching them up, we're talking about sliding them across each other. So why does this happen? So the decision to move is activated by the brain. The signal is transmitted down the spinal cord to the motor neuron, which you see there in yellow, and the motor neuron touches the muscle. So the neuron and the muscle are both activated or stimulated by what's called an action potential. Now, an action potential is a change in voltage across the membrane. What do I mean by voltage? I mean a change in the ion concentrations of predominantly sodium and potassium across the plasma membrane. And changing those ion concentrations is like electricity. It's called an action potential. And it's going to change the voltage across the membrane. So we have to get from the brain down the motor neuron. The motor neuron rests on the muscle fiber. We have to get that signal from the brain down the motor neuron. From the neuron, jump over onto the muscle fiber, down into the muscle fiber so that those things can slide across each other. All right, when we want to change the membrane potential, when we want to change how much sodium is on one side or how much potassium, we have to talk about ion channels. Some of them are chemically gated, meaning if a chemical arrives, it will open. If the chemical's not there, then it won't. And so that's going to be one of the ways that we're going to move sodium initially. When there is a chemical messenger called acetylcholine, acetylcholine touches that chemical gate, opens it up, and we have sodium passing in. Once sodium passes in, it changes the voltage. Then we can switch to the voltage-gated ion channels on the right-hand side. When the voltage changes, that thing opens and more sodium can go in. All right, so we'll look at this in a little bit more detail. As far as the motor neuron, as far as the neurological component, when we're talking about somatic motor neurons, somatic motor means voluntary. And so that's anything that I can thoughtfully and conscientiously do, that's going to be a somatic motor neuron. So if I'm going to think about contracting my bicep, somatic motor. Motor because it's movement, somatic because it's voluntary. Axons. So when we talk about a nerve cell, we looked a little bit at nerve cells in chapter 4. It had that big cell body and those extensions. The long thread-like extension is called an axon. So we have the cell body and the axon is going to land right on a muscle fiber. So this is called the neuromuscular junction. When we look at this picture, the axon in yellow is the end of a neuron. You can see faintly underneath that white wavy line, the sarcomere. You see the red myosin fibers, you see the blue actin fibers, right? And so you can see how we have a neuron sitting on the surface of a muscle fiber. A lot is happening here. So we have the sarcolemma in white, and you'll notice it comes from the right, it weaves and goes, and then it goes off to the left. What you'll notice on that sarcolemma are those little blue, right, coming back here, chemically gated ion channels. This is going to be important because what's in those vesicles? Inside the axon, right, see the word synaptic vesicle with acetylcholine? So when the message goes from the brain down the axon, all right, it's going to trigger that acetylcholine, and you'll see that at the bottom, to release into that area called the synaptic cleft. The little greenish yellow dots bind to those chemically gated ion channels, so they open up and sodium can move. Fascinating. Still haven't talked about calcium though, have we? We're getting there. This way, how about that? So the big picture, this is what needs to happen. We need to think about it. We need to have events happening from here, the neuromuscular junction. We need to excite that muscle fiber so that it can send that stimulus down through that white, those T-tubules. Then we have to get excitation, contraction, coupling, right? And then cross-bridge cycling for contraction. So this image on the right is a really great, simple overview. It's great. All right, it says the motor neuron fires an action potential down the axon. We don't really know what that means yet, but we will. The motor neuron's axon terminal releases acetylcholine into the cleft. Acetylcholine binds to those receptors on the junctional folds, those chemically gated ion channels. The binding causes a local depolarization called an end plate potential. Depolarization means sodium goes in. Muscle fiber excitation. The local depolarization then travels along the sarcolemma and down into the T-tubules. Sarcoplasmic reticulum releases calcium. Calcium binds to troponin, which moves the tropomyosin, opening up the actin binding sites. Myosin can now attach, and those fibers can cross each other. Did you lose your mind yet? I think it's time. You need a deep breath. All right. So we're going to go through each of these steps. Now, the action potential arrives at the axon terminal. And basically an action potential is a change in voltage across the membrane as a result of the change in protons, sodium ions across that membrane. So the action potential arrives at the axon terminal. The calcium channels open. So that change in voltage, that sodium influx, causes calcium channels to open. Calcium enters the neuron. Calcium causes those acetylcholine vesicles to release acetylcholine. Acetylcholine binds to the channel receptors, opening them up, right? Sodium chemical gates. Sodium is going to enter the muscle cell, resulting in what's called an end plate potential. In the cleft is something called acetylcholinesterase. It's an enzyme that degrades acetylcholine because we don't want constant contraction. We only want contraction when we want it. So we want to say, okay, yes, contract, now stop contracting. Once I've decided no, the signals stop, acetylcholine should go away. That's what acetylcholinesterase does. So we have the change in action potential down the neuron. We have the voltage-gated channels. Calcium comes in, right, to that yellow bulb, to the end of the axon. Acetylcholine is released into the cleft, the yellow dots. The yellow dots attach onto the sodium gates. Sodium gates open, sodium goes in, and that triggers the voltage-gated channels all along the membrane. This would be a great video to watch. How long is it? Three minutes? Alrighty, let's watch. Okay, I guess we're not watching it. So go down to go to your mastering and find the neuromuscular junction A&P flicks. All right, good, very, very good video. All right, so why is this important? Why do we want to know this for anatomy and physiology? Well, some things, toxins, drugs, and diseases, interfere with events at the neuromuscular junction. General anesthesia is about stopping, right, any kind of nerve transmission to the muscle. Myasthenia gravis is a disease, an autoimmune disease that has to, that involves acetylcholine receptors. So knowing about this microscopic anatomy and microscopic function of muscle cells can be important. All right, so now that we've gone from the brain down and now we have the channels open, right, acetylcholine got on there, sodium, boom. This is called the action potential. The action potential is where we talk about the movement of sodium and potassium to send that signal down the muscle cell. So we sent one down the nerve cell, we jumped the cleft with the acetylcholine, now we want to send that message down the muscle cell. How does that happen? So when acetylcholine binds to the ACH receptors, sodium diffuses in. Sodium diffuses in, which makes the inside of the cell more negative, less negative, more positive. And that causes in a depolarization event. The depolarization event causes those voltage-gated channels to open. Notice how more sodium comes in. A large influx of sodium triggers the action potential, and it's unstoppable. Once we have jumped the cleft and we have opened those voltage-gated sodium channels, we are contracting. Nothing you can do about it. So the action potential is traveling, that big pink arrow, down the sarcolemma from one voltage-gated channel to another, causing that area to depolarize. The other thing that's important is that this depolarization is going to go down the sarcolemma and also down those T-tubules, right, which we'll get to in a minute. Once the action potential travels past, we end up with what's called a repolarization event. Repolarization event is when the sodium channels close and the potassium channels open. And we want to have that sodium leave the cell because sodium is a positive ion, a cation. With the sodium came in, Na+, potassium goes out, K+. They're both positive ions, but we have to establish that resting membrane voltage if we want to be able to have a contraction, another one follow it up. All right, and then something called the sodium-potassium pump is going to reestablish those concentrations. This is what a action potential tracing looks like. Sodium channels open up. We have a lot of positivity. Notice on the right-hand side, it goes from -90 all the way up to plus 30 because inside the cell is negative. We open those gates, all of those positive ions flood in, the inside of the cell starts to become very positive. That's your depolarization event. Once we hit that equilibrium point, right, the sodium channels close, the potassium channels open, and the positive ions begin to leave, and we'll notice that the charge goes way down again. That's an action potential. So now that we have followed the action potential through the movement of sodium across the sarcolemma, it's going to go down through the T tubules. Now, the T tubules lead to the sarcoplasmic reticulum, which is very sensitive to voltage. And when the voltage change happens, the sarcoplasmic reticulum releases calcium. And so once that calcium is released, you can see here, it's called excitation contraction coupling. And we have, if you look really closely, you see the axon terminal, you see the little tiny acetylcholines, you see the action potential go down, right, down along the sarcolemma. You see it go down through the T tubule, and then you see the little red dots. And the little red dots are calcium. And calcium is now being flooded into the muscle tissue itself. Well, why do we care? Remember back to when we had the red myosin and the blue actin. What we want is for myosin and actin to be able to connect. When they connect, they can contract, right? But if they can't connect, they can't contract. So calcium, what it does, if again you look really closely, you can see that little yellow troponin molecule and that tropomyosin strand. Calcium is going to bind to troponin, and it's going to move that tropomyosin strand. And what that does is it opens up the actin binding site for the myosin. All right, so without calcium, we cannot have muscle contraction. This is the excitation contraction coupling. The excitation of changing that membrane potential down the T tubule, releasing all that calcium. Calcium binds to troponin, which pulls the tropomyosin. The myosin and actin bind, and we have contraction. Again, muscle contraction part 2, excitation contraction coupling. Go find it, go watch it. This is confusing stuff, right? Make sure you are doing all the things you need to do to make sure that you can follow this. So here we are. Tropomyosin blocks actin, but when we have calcium, all bets are off. At higher intercellular calcium, it binds to troponin, moves tropomyosin, and allows myosin to bind to actin. Now, when they bind, right, it's called cross-bridge cycling. Right, and we're going to go to the picture. It's a little easier to see. So number 1, cross-bridge formation. You see the calcium has pulled that yellow line away from the big blue dots, has pulled tropomyosin away from actin. Now you'll see that the myosin is attached. You'll see this little energy there. And you'll notice how it goes from, I can't do my hand like this. It goes from like this, and it goes like this. See in picture 2? It grabbed onto the actin and moved it forward. All right, that's called the power stroke. Grab the actin, moved it forward. Now it detaches because now the pressure is too great, right? We've moved it forward and the proteins can't bend that way. So the ATP, boom, and now we're back. And then we're going to grab it again and move it forward. Bend down, go back, grab it again. So it's like a tug of war. The myosin grabs and pulls, lets go, grabs and pulls, lets go, grabs and pulls. And this only happens as long as calcium is there. Calcium is not there, right? It's going to close those bad boys up. So this is part 3, the cross-bridge cycle. Go watch it. Won't be disappointed. Bring popcorn. All right. So if we want to talk about the entire thing from start to finish, right? We probably should do that right now. The action potential starts at the brain. It travels down the neuron, down the axon to the axon terminal. The axon terminal, the action potential, causes calcium to flood into the axon terminal. Terminal. Flooding of calcium into the axon terminal causes acetylcholine to be released into the synaptic cleft. The acetylcholine binds to the chemically gated sodium ion channels on the sarcolemma, opening them up. Sodium starts to flood in. We're going to call that depolarizing. When we change from that -90 to +30, when we really increase the number of positive ions in there, it triggers the voltage-gated channels all along the sarcolemma to let in more sodium. That sodium travels down this, that action potential travels down the sarcolemma, down the T-tubules. When it goes down the T-tubules, again, voltage-gated channels, sodium everywhere. It triggers the sarcoplasmic reticulum to flood the area with calcium. Calcium then binds to troponin, which moves tropomyosin, which allows myosin to attach to actin, and contraction happens. At the same time, we only want contraction to happen once. When we're done, the lack of the voluntary somatic motor action potential allows for acetylcholinesterase to degrade the acetylcholine, closing those voltage-gated and chemically gated channels. The sodium-potassium pump reestablishes the sodium and potassium balance. Calcium is resorbed by the sarcoplasmic reticulum. If calcium is resorbed, it gets pulled away from the troponin. Tropomyosin covers the actin binding sites, and we have relaxation. It's a lot. It's a lot. We will definitely review this in lecture when we're face-to-face, because this is a big one. All right, so that's the microscopic stuff. So let's talk about, what actually happens when we contract a muscle from a whole muscle standpoint, not just the cross-bridge cycle at the microanatomy. We want to produce tension. So we can do tension or force in a couple of different ways. Isometric contraction is if I just do this and go like that. I just tightened it, but I didn't shorten it. Isotonic means I shortened it. When we talk about muscle contraction, right, the force and the duration of contraction are going to vary. And it's going to be based on the stimuli and the amount of frequencies and intensities. If I want to hold up this pencil, that's one stimuli for my bicep. If I were to try to lift up this whole desk, that would be a lot more force required, right? Each muscle is served by one motor nerve, and a motor unit is the nerve muscle functional unit. When we think about a motor unit, right, so you have a little cross section of the spinal cord, you have the axons reaching down and touching those muscle fibers, and you'll notice that motor unit 1 and motor unit 2 are going to be responsible for contracting different muscle fibers. Sometimes we'll need them all, sometimes we won't. If I'm just holding the pen, I might only need motor unit 1. If I'm holding the pen in the computer, I might need all the motor units. So a motor unit is a motor neuron and all the muscle fibers it supplies. The smaller the number, the greater the fine control. So the motor units that feed the little fingertips that we're trying to do little things like thread needles, very fine control, small motor unit. If I'm trying to lift up this desk, right, with my shoulder muscles and my arm muscles, lots and lots and lots of fibers. This is a picture of a muscle twitch. Do not confuse this with the action potential diagram that we just saw. This, if you read the x-axis and the y-axis, is how much time it takes to achieve maximum tension. The other one was millivolts, right, versus time. Millivolts is measuring the electricity, the voltage across the membrane. This is a muscle twitch, totally different. This is pressure. All right, so we have the latent period, the period which nothing's happening. See that like aqua blue stripe? It looks like nothing's happening. But remember what we just talked about. Everything from the brain all the way until actin and myosin go like this. It's a lot. Action potential, sarcolemma, acetylcholine. That's all happening in the latent period. Period of contraction is the period of this, when we have actin and myosin and calcium and tropomyosin and troponin actually interacting. When we pull that acetylcholine away and we pull the calcium back, we have our relaxation period, where these two things fall apart. The elastic fibers allow for recoil. So this is more of a physical picture of what's happening during muscle contraction. Now this is 3 different muscle twitches depending on the muscle type. So the eye muscle is very fast, like boom, 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 right? We can move our eyes left, right, up, down, quick, 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 quick. So the myogram is going to be up, down. The gastroc is a calf muscle, a little slower, but you can go up, down, up, down. The soleus contraction, right, takes a little bit longer. muscle twitch. It has to do with the metabolic properties, what that muscle is meant to do, right? Like anything else, structure, function. Then we have graded muscle responses. So the normal muscle contraction is very smooth, and how much muscle is called to action varies with needs. A muscle twitch is only a thing seen in a lab, not something we can normally produce, right, in our own body. Normally we have what's called a graded muscle response. Changes in the frequency of stimulation and the strength of stimulation. So how often, and then how strong. And it's just going to change how we move our muscle based on what the muscle needs to do. Do I need to kick a ball? Do I need to run? Do I need to lift this up slowly over my head? Do I need to throw it? Right? Frequency and strength. That's how we gauge our graded muscle responses. Most of the time when we're doing muscle contraction, we're not just going to do a twitch. We're going to do a summation of some sort. This is temporal summation, where we stimulate, relax, stimulate, relax. They don't completely relax. And this isn't really a natural thing. We're getting there when we talk about unfused tetany, smooth, continuous contractions, high stimulation frequency, boom, 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 boom. Most of the time, if I'm going to take something like my water glass and hold it, right? I'm going to hold it. I'm not going like this. I'm just holding it. That's when we get to what's called fused tetany. It means very smooth, sustained contraction. All right, this is what we usually see with our muscle, our graded muscle response. We don't see a lot of jerky movements, right? We usually get to what's called fused tetany. Again, coordinated, smooth, sustained contractions, depending on how heavy the thing is and how long we have to hold it. Now, the more force we need, right? If I'm carrying the pencil versus carrying the whole desk, I'm going to need more motor units, right? So there are different number of motor units called to work. It's called recruitment. Recruitment means we're going to use as much of the muscle as we need to get the job done. Isotonic and isometric, we already went over that. This is just a picture of it. Shortening versus just tensing the muscle. And then the last part of the chapter, I think, or the last part of the discussion on skeletal muscles is where we're getting the energy. So we're getting our energy from ATP. We are also getting it from creatine phosphate, and then either anaerobic or aerobic mechanisms. So if we're creating ATP from ADP and creatine, very quick, direct phosphorylation gives us enough energy for about 15 seconds. So jumping, running, hitting, throwing, something fast. The anaerobic pathway, right? We're going to go through glycolysis, and then we're going to take a right turn and convert to lactic acid. Anaerobic is basically happens when there's no oxygen present. An anaerobic example is if I'm going to go start doing biceps, and I'm going to curl and curl and curl and curl. The more I squeeze my bicep, right, the more I'm preventing blood from entering the muscle tissue. If I'm preventing blood from entering the muscle tissue, I am reducing the sugar, reducing the oxygen. Well, I'm still trying to contract my bicep, so my muscle says, well, we need to go anaerobically. Then we're going to build up that lactic acid. I'm going to feel that burn later, right, in the meat, right? So anaerobic, no oxygen, but does create lactic acid. Only 5%, right, but we can still do more with anaerobic pathways than we can with direct phosphorylation pathways. Aerobic is where it's at. 95% of our ATP at rest and light to moderate exercise. It's slower to get going, but way more efficient. We're going to use our sugars, then we're going to use our fatty acids. And this is a really sort of the workhorse of creating energy for contraction. So this just shows a woman who is running. She's either doing a sprint in the blue or more of a sort of a marathonish type run, prolonged duration. And you'll notice how the first 6 to 10 seconds we have ATP stored, ATP created from creatine phosphate. 30 to 40 seconds we have our glycogen, and then we end up with a little bit, right, oxidized anaerobically. That was quick. It only took like 40 seconds. If we want to do hours and hours and hours, right, we're going to go through the aerobic pathway. So we have different methods to create ATP depending on what we're going to do. The force of muscle contractions depend on the number of muscle fibers stimulated and the relative size of those muscle fibers, right? The more, the bulkier the muscle, the more tension it can have. And this happens when we work out, right? We can increase our muscle size based on what we're asking it to do. Let's skip some things. So factors that increase the force of skeletal muscle contractions: frequency of stimulation, the number of muscle fibers, the size of the muscle fibers, and if we stretched them first and then contracted, get a little bit more out of it. So frequency of stimulation, number of muscle fibers, size of muscle fibers, and if we stretch first, we can, because we're pulling those farther away, and then we can really have a lot more cross bridges. All right, let's talk about smooth muscle a little bit, and then we will end this muscle chapter. So let's stop, put skeletal muscle aside, and let's talk about smooth muscle. Found in the walls of most hollow organs: respiratory, digestive, urinary, reproductive, some of our arteries. Smooth muscle is packed into sheets. Now, when we talk about smooth muscle, we talk about these spindle-shaped cells. They're going to come in 2 layers. They are going to have some connective tissue sheets around them. We have what are called varicosities instead of neuromuscular junctions. So we don't have that axon, terminal, that whole thing. We have what's called a varicosity. Let's take a look at that. So each one of those cells, you'll see this little, so the red kind of bent shape up there. Each one of those cells is one smooth muscle cell. Each one has one nucleus in it. But you'll notice that it's has that autonomic nerve fiber. It's one nerve fiber, but notice how it branches out. It almost looks like seaweed and has these little varicosities, similar to the acetylcholine vesicles that we had before from the axon terminal, except it touches all over the smooth muscle cell. And this is to ensure that when one nerve fiber gets stimulated, that entire area will contract at the same time. Has a lot of sarcoplasmic reticulum, no T-tubules, but we're still talking about calcium. We're still talking about actin and myosin. All right, it's just a little bit of a different setup. We have calcium, right, activating the calcium channels. Then those calcium channels, right, are going to flood the calcium along the membrane of the smooth muscle cell. We still have that thick and thin filament thing. Thick filaments are fewer, right, and have myosin heads along the entire length. There's no troponin, that we're not worrying about it. Instead of troponin in smooth muscle, we have something called calmodulin. So we have calcium binding to calmodulin, moves the tropomyosin, the actin and the myosin attach each other. Some things are similar, all right, but some things are not. This table 9.3, again, talks about the body location, cell shape. Then this part talks about T-tubules, yes or no, myofibrils, yes or no, sarcoplasmic reticulum, gap junctions, neuromuscular junctions, how it's regulated. This is a great table, right? Source of the calcium, and then how these things work together. Notice how the troponin actin combination works for skeletal and cardiac, but not smooth. Notice how the myosin and the calmodulin, right, the calmodulin regulates the calcium. So take a look at this because this is really important. This will give you all kinds of great information on the comparison between the 3 different types of muscle tissue. When we talk about smooth muscle tissue, how are these, what's the mechanism of contraction? Because when we talk about skeletal muscle, we know we have to think about it. Here we have smooth muscle is autonomic, right? Not voluntary, involuntary. We have some self-excitatory cells, we have those varicosities, but it's autonomic. And the body will take care of monitoring, right, the activity of smooth muscle. Contraction in smooth muscle is different from the skeletal muscle. Calcium, some of it comes from the sarcoplasmic reticulum, but mostly it comes from the extracellular space. Calcium binds to calmodulin. All right, and then we have the myosin kinase, the light chain. So it's a little bit different, but has a lot of similarities. Calcium is important. Calmodulin activates the kinase, which activates the myosin, which allows it to attach to the thin filament. It's a little bit of a different step, but again, we want those thick and thin filaments to still interact. How is it regulated? Neurally, right, by graded action potentials down the varicosities, or it can also be response to stretch when your stomach fills up, when your bladder fills up, right? It'll start to contract. Oh, guess we're done. Hormonally, right? Hormones, different types of digestive hormones can cause your digestive system to begin to move and churn, right? That's why the smell of certain foods can make you hungry, because the smell triggers the brain. The brain says we could eat, and then it tells the body to start to churn. When it starts to churn, you think, I think I'm hungry, right? So smooth muscle is autonomic. It's subconscious, a little bit different. All right, so that's going to be the end of chapter 9. I know it's a lot, and I also know I speak really quickly. The good thing with videos is you can go back and look at the ones that you want to review. You can take a look at the images in your book. You can take a look at any of the videos that I have attached. And hopefully when we get into lecture, you can ask any questions and I can try to make it a little bit more clear for you. So thank you for watching chapter 9.